Okay, next we're going to our next speaker is called Adjunct Professor Scott. He's the acting director of scientific biology at CSIRO platform. Colin was awarded his PhD in molecular microbiology in 2000 from the University of Sheffield, UK, where he worked on transcriptional regulation and psychological adaptation in bacteria. He moved to Australia and joined CSIRO to develop enzymes that degrade detoxify pesticides and to study how they evolve. This works into developing enzymes and complex bio-based devices by catalysts. He now leads biocatalytic and synthetic biology team at CSIRO, a foundation technical application domain. Please welcome Professor Colin. Thank you.
Thanks. I'll just try and share my screen. So hopefully you can all see this. So look, thanks very much for inviting me to speak today. I very much appreciate it. As mentioned, I'm the acting director of CSIRO's Future Science Platform in Synthetic Biology. And CSIRO, for information, is Australia's national laboratories. And this Future Science Platform is our attempt to kind of build a cohesive community of practice in synthetic biology in Australia. Because when we started down this track about 5 years ago, there really wasn't much synthetic biology happening in Australia. And over the last 5 years, we've managed to build something of a community of practice here around SynBio and train a relatively large number of people to be experts in that area. So today I'm going to talk about a piece of work that we've done to transplant metabolic pathways for ex vivo operation in continuous flow. I'll explain what that means as I go along. But before I do start to kind of get into the science, I'd like to acknowledge the people who've actually done the work on this project and made it work. So for the work we'll talk about today, most of the molecular biology and protein biology happened in my lab with Carol Hartley and Nigel French, and Andrew Warden from my lab providing molecular modelling. And there's a fair bit of chemistry that had to go into this work as well, and that happened in Charlotte Williams' lab in Melbourne through Andrea North and Quentin Churches. So when we look at metabolism, and please don't try and see the detail here because you can't, the first takeaway message is it's really complicated. It also happens within a cell, and so there are mass transfer issues across the membrane of the cell as well. And as a biologist, I see this figure and I think, that's beautiful, I want to understand how it works. As an engineer, I look at this and I think, my God, it's so complex, how am I ever going to do anything with this? And so really we'd like to be able to simplify metabolism. In my lab, we want to be able to take just the bits of metabolism that we're interested in, to rebuild them ex vivo in test tubes, and to have them work together to do something useful. In this case, it's making chemicals. And obviously there are lots of labs that do this. It's called biocatalysis, and you can do it in a big jar and just put all your enzymes in the big jar and have them do their thing. If you do that, you don't have a great deal of control over what's happening. And so in recent years, we've been looking at moving enzymes into continuous flow. So continuous flow, essentially in this case, consists of a number of reactors that are linked together, and each reactor contains just one reaction, so just one enzyme catalyzed reaction. The enzymes stay in the column, they don't move into the next one, they're immobilized, and they conduct one reaction, and then the intermediate from that reaction flows on to the next one. It gives you a very high control over the reaction, and also limits the amount of purification that you need to do at the end of the work. So this animation has failed, so I apologize for that. There are a couple of things that you have to do to be able to adapt an enzyme to be able to work in a continuous flow system. So, as I described, you have a reactor, and inside the reactor you have your enzymes on beads or whatever the surface is. And so you have to be able to immobilize the enzyme onto the surface effectively in a way that it doesn't then leach out of the reactor. When you have enzymes that require cofactors, you also have to either provide a very large amount of cofactor that flows through the column and is lost, or you have to have a system whereby you can retain and recycle that cofactor, which is what we've done in this project, and I'll talk about that in a little bit. So, when it comes to retaining and recycling cofactors, we cast around nature. We have a look around nature to see what nature does in some of its systems, because there are a number of reactions that require intermediates in reactions to be captured and held and passed from one reaction site to the next. And so there's an example here of pyruvate dehydrogenase, and it has this long flexible arm that takes intermediates from reactions from one active site to the next. The same sort of thing happens with fatty acid synthase and with polyketide synthase as well. And so we took our inspiration here. Could we introduce long swinging arms into enzymes that could recycle their own cofactors by having multiple active sites, one for using the cofactor and then one for recycling? And so this was kind of the model, the blueprint that we came up with the cofactor trapped between two fused enzymes, one of the enzymes doing the reaction that you're interested in, the other enzyme doing the recycling of the cofactor. And between those two modules, we tether a cofactor on a long swinging arm that can reach between the two active sites and transport the cofactor from one place to the other, but prevents it from diffusing away into the medium that's being flowed across the top. And I'll try and describe a little bit more about that now. So really the kind of enabling piece of work that was done here was the modification of NAD, or ATP, in both cases here, to be able to accept that long swinging arm, which we, for this particular example, we used PEG. And so you can see that what we've done is we've modified the primary amine of the adenine moiety in both cases by activating that primary amine. We were then able to introduce the long swinging cofactor, which would then be used to retain the cofactor and allow it to swing between the two active sites. The chemistry here is actually really quite difficult, and it's a huge credit to Charlotte and her team that they managed to make this work. Of course, we wanted to make sure that the cofactors, once modified, still worked, and so this is just a piece of work showing that the modified NAD that we've made retains the same, or roughly the same amount of activity as unmodified cofactor. And you can see that there's a log scale on the y-axis here, but you can see that we still have very activity, all the enzymes that we investigated. The cofactor was then immobilized to the enzyme. So the enzyme is a fusion with two enzymes next to each other, fused genetically, so there's a single gene that encoded them. And between them, there was a linker, a polypeptide linker. In the middle of the linker is a cysteine, and at one end of this modified cofactor is a maleimide moiety. And that maleimide reacts with the solvent-accessible cysteine that's present in that flexible linker to covalently bind this modified cofactor to the fused enzymes. So that's looking at retaining and recycling the cofactor, and that works quite nicely. We then wanted to be able to immobilize the enzyme to the surface. And as you can imagine, this is already quite a complex piece of biochemistry that we've put together. And we didn't want to just immobilize by cross-linking, because that would inactivate a very large proportion of the enzyme that we want to go onto the surface. We really want to retain as much activity as we could. And so we looked at site-specific immobilization technology, specifically using the formation of a covalent bond to the surface. And to do that, we've exploited esterase chemistry. So this is effectively a carboxyl esterase shown here, and it performs a fairly simple reaction where it hydrolyzes using water to break a carboxyl ester bond. It's a very well-known reaction. The mechanism is really well understood. But one thing that we do have access to are inhibitors of these enzymes that form covalent bonds with the active site serine and irreversibly inactivate them. And so we exploited this in our immobilization technology. So what we've done is we've taken some fairly standard divinyl sulfone beads, and we've modified them to have a trifluoroketone on the surface. And this chemical is an irreversible inhibitor of an esterase, and it forms a covalent bond then with the enzyme, and because it's on a bead, the enzyme is irreversibly bound to the bead. And so the next thing that we did is we fused this enzyme to our already very large fusion in the same way. So now there's a single gene. This single gene encodes the enzyme that does the process that we're interested in, the recycling enzyme and the immobilization enzyme, with the cofactor tethered covalently between the two active enzymes. So the question we have now is, does the design work? And I wouldn't be talking about this if it didn't work, so it works quite nicely. We selected a reaction for our prototype, and this reaction takes a fairly cheap and abundant chemical, just glycerol, and it converts glycerol through to an anti-diabetic compound. And this is a cascade that involves several steps. One is ATP dependent, one is NAD dependent, and we have enzymes that are modified with covalent bound ATP and NAD in those reactors. And we also use an enzyme that forms a carbon-carbon bond to form this dihydroxy ketone intermediate that can be very simply chemically treated to cyclize. So this is the prototype reaction that we chose, and those are the enzymes involved. And we tested each of the reactors to see if they worked, and they worked very well. One of the things that you'll notice here are the total turnover numbers. So that's the number of, on average, how many times a cofactor can be reused for a reaction within the reactor. And so we're looking at numbers in the range of 10 to the 4, which is reasonably high. So for every one of the molecules on the surface of the reactor, we can make around about 10 to the 4 molecules of product. The space-time yields are also very good. This is Carol Hartley. We repurposed an old HPLC, an old protein purification kit, to run our first reactors. And you can see there are columns there. The columns contain the DVS beads that have been treated with our enzymes, and we have flow from one into the other, with the graph on the left here showing that we're getting very high conversion rates, so almost all of the glycerol is consumed and converted into this anti-diabetic drug. So we were reasonably happy with that result. So in summary, we have a generalizable chemogenetic engineering approach that allows cofactor retention and recycling. We've demonstrated it with ATP and NAD. We've also demonstrated it with NADP, but I haven't shown it here. We've also developed a highly efficient orthogonal enzyme immobilization technology that makes covalent bonds that means the enzymes don't leach out of these reactors. We've shown with a couple of cascades, I've only shown you one here today, but with a couple of different cascades, we've shown that we can make some quite complex cascades, and we hope to increase the complexity of those so that we can have very efficient reactor systems without purification between steps. Substrate conversion exceeds batch, and I didn't show this today, but in batch reactions, the cascade that we built here is subject to product inhibition from the product of the reaction, which means it accumulates in the batch reactor. It doesn't accumulate in a continuous flow reactor, and so we don't have feedback inhibition or product inhibition in the system, and so we can get very, very high rates of conversion that we just don't see in traditional batch reactions. And the total turnover numbers, the number of times each cofactor is used, is higher than competing systems. So it's a very efficient and effective system. With that, I'll say thank you very much for your attention. Very much appreciate the opportunity to talk here today, and I will leave it there. Thank you very much.
Thank you very much, Prof. Colin. It's been really great hearing from you. I happen to transition from how frugal hardware could be used for enabling synthetic biology and other applications on the continent, and also just hearing from you how you are leveraging biology in solving problems in Africa and elsewhere. We're going to hear again from another very interesting speaker who is also going to speak to how he and his group are using frugal open source hardware to overcome challenges to doing synthetic biology. And he's no other person than Dr. Vamsi. Now, Dr. Vamsi is an interdisciplinary researcher with an academic background in synthetic biology, biophotonics, and hardware for biomedical applications. He has a special interest in bridging the gap between technology and the life sciences. He received his PhD from the University College Cork. He has a working experience in computational designing and modeling proteins with special optical properties for targeted therapeutics and diagnostic applications. He also has vast experiences working with teams specializing in artificial intelligence, machine learning, virtual reality, photonics, and hardware. He enjoys exploring ideas focusing on technology development into life sciences applications. Let's welcome Dr. Vamsi to give us his presentation. Thank you.
Thank you, everyone. I hope you can hear me all right. And I hope my screen is visible as well. Okay. So today I'm going to talk about deployability by design in synthetic biology solutions. And there's a reason why I put the word called solutions in there. So I've been tracking synthetic biology since the last 5 years. And in my PhD, I've been working on projects that revolve around deploying solutions, not just a part of technology, but an entire solution. So throughout the SynBio Africa conference, there have been very interesting speakers and very interesting topics. And one of my colleagues who just presented just before me, China, raised a really good discussion on low-cost hardware as well. So let's get back in here with a technology what he introduced in before. So this is a technology what we worked 3 years before. China has introduced that to you. So in a traditional way, you can see a biologist doing optical growth monitoring a lot. So in this growth monitoring, people used to take a bacterial sample, put it in a falcon tube, throw it into a shaking incubator, and then take it into a laminar airflow chamber, and then take out a sample and get it into a spectrophotometer to get an optical density reading. Now, this is really a pain for every biologist because you do this every 20 minutes or 30 minutes, and every time you have to stop your experiments and go in there to do this, and there's a huge lot of consumables that's going into the waste, and then there's a lot of power loss during this as well. So how about thinking it in a different approach? Why do we have to take these into a shaking incubator? Why do we have to go into the hood? Why can't we redesign the technology? And that's where the small idea of using a fitness tracker came in. This fitness tracker has all the components that are required to do the same optical density monitoring. If you look at it in its simplistic terms, light has to pass through a sample which has a different optical density, and then a detector has to detect the amount of light that has been absorbed or transmitted later. So that's what we did in here. We hacked the fitness tracker in there and modified it to wrap it around the falcon tube where the bacteria is being grown, and the data is continuously relayed to a smartphone. And that is a 3D printed enclosure around it. You can see it's a small little handheld device, and you can see a small euro coin right beside it for comparison of size. Now what happened is, this changed the way we worked. You don't need to go into the hood anymore. You don't need to go every 20 minutes. You just lodge this tube into a holder, throw it into a shaking incubator, and the readings are automatically taken. So there is no risk of contamination either here because you're not opening the tube, and the consumable problem is also solved. Now, a critical thing to see here is, although the traditional spectrophotometer is very versatile and it can do a lot more things, now designing a solution for a problem, it took more than what it does by the spectrophotometer. So we designed a solution where you can get rid of the plastic, where you can get rid of the movement in there, where you can get rid of the sampling in there. So this is very important, and this we could be achieved only because from the beginning we were thinking about how to deploy the solution. We even got to design a mobile app which collects the data. We tested it on multiple bacterial strains, and we published this article a few years ago. And then we went into the next thing. How can we make it more versatile? What more features can we add into it? Can we throw fluorescence into it? And if we throw fluorescence into it, what will we use this device for? Should we use it for a point-of-care device? Can we measure any other samples apart from bacteria? Can we measure water quality with this? So these are the type of questions we've been battling through when we were building these handheld gadgets. Now let's jump on to protein-based technologies, right? So you can see a small schematic in there. The small little green part there is the natural proteins that are existing, and the yellow part are the fusion proteins. And you can see 95% of protein engineering till date is done by this method. But then the whole canvas of de novo proteins came in a few years ago by David Baker's paper, and then this just expanded the field enormously. And since then, you could see a lot of protein companies coming in the design phase or the build phase, the test phase, and even the product phase. And these startups are trying to commercialize a lot of protein-based products. And what's interesting here is, although there are small companies always coming through, big pharma and big biotech are also very interested into it because they see the potential. Now, this boom in protein-based technology is just an example how the synthetic biology is expanding. A lot of startup companies and a lot of promise for future indicates that this field is ready to deliver. Now, when we think about synthetic biology products, we are solving the problem in here. And solving that problem is taking a solution as a set. And how do we deploy these things? Most of the scientific things are based on laboratory-based technologies. How do we get them onto the field? And that's where the deployability comes in. And when we talk about deployability, we're talking about here feasibility, safety, and regulations. And the feasibility can be subdivided into technological feasibility and the commercial feasibility of the product. And then how safe it is to bring it outside. Does it have GMOs? Does it leak? Does it contain everything inside? And then the regulations would come in. And then we should also be thinking about the mode of deployment. Is it going to be off-grid kind of deployment? Is it point of care? Or is it at home, near patient? Or is it lab-based? Or is it going to be embedded into an industrial setting? These are some of the thought process we were going through as we are designing these solutions using synthetic biology and low-cost hardware together. Now, if you look at a biosensor hardware, we all know it contains a sample or an analyte, and then a biological receptor, a transducer that will detect, and then a detector to detect the signal, and then a smart device like a smartphone and an analyzer to analyze the signal in there. We've seen many examples of off-grid devices, sensors in agriculture plants, wastewater treatment plants, and you can see at-home tests such as pregnancy kits and antibody-based COVID kits, which are coming out these days. And qPCRs and blood profiling are good examples of laboratory-based testing, and classical process monitoring is the industrial-based settings. And in each of these newer devices that are coming through using synthetic biology, the deployment mode should be taught in the design phase in order to see how the model is going to be looking like. So in the previous talk, Chinna has briefly introduced about some of the devices that we are working on, whether it be ODX, which is the optical density monitoring, or the ColorX, where you're talking about detecting color changes, or the mobile fuse, or even the mobile lamp in there. All of these are very simple instrumentation made with frugal hardware using commercially available parts and technologies. So the innovation comes in here in how are we thinking of deploying these technologies, and that determines the shape and a lot more features. Now I'll give you a brief, very small examples. Suppose we're trying to build a sensor or a technology for off-grid sensor. So feasibility features such as, is it portable? Can it be mass manufactured? Is it weather resistant? Can you put it anywhere without worrying about calibration? Is it easy to install and easy to use? Is it battery powered, so that you don't need to have any cables? And does it need many maintenance and report? Can it wirelessly lock the data? These are some of the features that we should be embedding into our design phase of the product before going and doing everything. And the last thing I would like to just put key points in here is that in synthetic biology solutions, we are now at an industry with a very early stage, and we have a lot of promises in here. And we need to make sure that the safety and sustainability comes in first. So how do we handle the samples or reagents? And what happens to the product after it is manufactured? How do we reuse and recycle? These are very important factors that we need to consider in the deployment of the design. Because in the last few decades, the chemical industries and many other industries have been at the similar stage, and they have not put the earth first, and we've seen the consequences what are happening now. Now, since synthetic biology is in a very early phase, it is time for us to think in a safe and a sustainable way and put these all topics into the design phase so we don't go too far without thinking these first. Now, coming to the key points, so as I was talking, we should be thinking about synthetic biology as a whole solution, not just as a particular sensor or a hardware or something. And then we have to think about the safety and product life cycle, and also in terms of developing economies, such as I come from India, and then we have Africa in here coming in here. And it presents a lot of opportunities because of the low-cost sensors and the synthetic biology decreasing the cost of DNA. So the field is booming, and it is time for us to think about deployability in a serious way. Thank you very much. And I'll leave you with a picture of some proteins that I've designed. Thank you.